Welcome, and thank you for joining us for today's webinar, Beyond Organic Acids. I would like to welcome our speaker for today, Dr. Betsy Redmond.
All right, thank you. So I'm going to go over organic acids and gut health. So I'm really looking at how can metabolomics identify what's going on in gut health. So it is organic acid. It's also going to be some amino acids, so plasma and urine. Let's get started. So metabolomics can provide information of metabolites produced from the gut microbial mammalian co-metabolism and is improving the understanding of molecular mechanisms underlying multi-level host microbe interactions. So essentially that's saying that we're starting to look at what are our gut microbiota and us talking about. So kind of eavesdropping into what they're talking about and what's that mean to health. I mean, normally the gut microbiota, pretty much always, is confined to the gastrointestinal lumen. So it's not going to make its way out. That'd be a problem. So in order to overcome this barrier, it's developed all sorts of tactics of releasing metabolites from different chemical classes to affect host organs to kind of get its business going. So we're going to go over that. So this is a nice diagram, and it looks at, a level for diet, a level of the microbiome, and then the metabolome, so all the products that are made by the microbiome, and the epithelium. So that the direct interaction between dietary compounds and gut microbiota are going to include the gut microbiota can transform dietary compounds into other products. And dietary compounds can modulate the composition of the gut microbiota. So certainly the microbiota you're going to see in a vegan compared to somebody on a carnivore diet is going to be different. But also that dietary compounds can modulate the metabolites that you'll see from the gut microbiota. So bacterial fermentation of colonic protein is referred to as proteolytic fermentation or metabolism. And carbohydrate fermentation is called saccharolytic fermentation. Fat is going to get absorbed in the small intestines and primarily is going to be impacted by bile salts that get secreted. So proteolytic metabolites have been implicated in metabolic disease like obesity, diabetes, colitis, non-alcoholic fatty liver disease. So we're going to be looking at those associations. So just an overview of protein fate. So protein comes in to the microbiota through two different ways. The majority of protein that reaches the intestines gets, most of it's going to get absorbed in the small intestines when you eat. About 6 to 18 grams a day of proteins or amino acids are going to reach the colon. So protein delivered to the colon comes from two sources. And you can see in this, it comes from endogenous or exogenous intake. So endogenous proteins are going to include things like glycoproteins or mucus, pancreatic secretions. And so it's going to vary also on health. So if somebody has a lot of inflammation or ulcerations, they're going to have more of these endogenous proteins that are going to be coming. And exogenous, you're going to get it from diet. So excess protein or incompletely digested dietary protein is going to make up that second component. And there's going to be several physiologic factors that can impact that. So GI factors like, PPIs or antacids. So maybe you're impacting digestion. How is digestion going? Also, if there's rapid or slow transit time, that can affect the microbiota. So protein itself, like how digestible is it? and digestion overall we're going to look at. The percentage of protein intake generally is going to end up in the colon. So if you have a really high protein intake, you're going to have a high overall level, more likely in the colon. Also, if it's mixed with other nutrients like fibers. So the microbiota can use the protein to do, like for proteolysis, to incorporate it into amino acids or structural proteins. But most of it's going to get fermented, and it's going to get fermented to different products and excreted. So tyrosine and phenylalanine are generally going to be fermented to phenolic compounds. Tryptophan is going to get fermented to indolic compounds, and then sulfur amino acids are going to be fermented to produce more sulfur compounds. So if we go here and we're going to look at microbial metabolites like amino acid metabolites. So high levels of aromatic amino acids, that's phenylalanine, tyrosine, tryptophan, and fermenting bacteria. So the bacteria that like to ferment protein. Two big ones are Clostridium and Bacteroides. So they favor greater production of phenolic and indolic compounds from these amino acids. There's a known positive correlation between fecal levels of tyrosine and phenylalanine and 4-hydroxyphenylacetic acid and P-cresol, and between fecal tryptophan and indole acetic acid. So we know that they are making those. Amino acids are going to require more interconversion steps for incorporation into biochemical pathways in comparison to monosaccharide units. Or carbohydrates. And it's not typical for the gut microbiota species to have the capacity to ferment all the amino acids. So amino acids are not generally considered as efficient an energy source for gut microbes as carbohydrates. So that's no surprise that gut microbes prefer carbohydrate. So this is an overview of phenylalanine tyrosine breakdown and all the things that it does. It can make melanin and thyroid hormones. It has 4-hydroxyphenylpyruvic and homogenestic, DOPA, dopamine, norepi, epi. So it has all these things it can do. But let's concentrate on this upper part. So when we look at this, we can see that 4-hydroxyphenylacetic acid comes directly from bacterial fermentation, and it can go to make P-cresol. There's also other ones to look at. 4-hydroxyphenylpyruvic acid, that can be involved in that bacterial activity, and so can phenylacetic acid. So when I'm looking at 4-hydroxyphenylacetic acid metabolism, it can come, it's a lot to say, it can come from kind of two ways. It can come from tyrosine directly, so bacterial action on tyrosine. So it's been attributed to several species, like I had said, Clostridia, Bacteroides, but also Roseburia, Bifidobacterium, and Fecaliprodsnitzi, as well as Klebsiella, Pseudomonas, and Proteus. And if C. difficile is around, it'll further decarboxylate it to P-cresol. So that can come directly from tyrosine. Otherwise, 4-hydroxyphenylpyruvic made from tyrosine can help build up more of the 4-hydroxyphenylacetic acid, as can tyramine. So tyramine is a naturally occurring trace amine derived from tyrosine that can be produced by certain gut bacteria. And then once it's produced, MOA enzymes can break it down to 4-hydroxyphenylacetic acid. So there's a lot of things that can go to make this compound, and most of them are going to come from tyrosine. They can also come from like polyphenols, green tea, quercetin, components of a Mediterranean diet. And that's, a lesser impact, but still that's one that can have an impact. So when I'm looking at this compound on a test, 4-hydroxyphenylacetic acid, this is an older study from 1979, and it was in infants who can have some pretty significant differences in gut bacteria and function. But they found that looking at their 10 patients, that those who had increased 4-hydroxyphenylacetic acid tended to have more abnormal issues, like they had giardia or an ileal resection or other diseases of the small intestine. And they concluded that measuring this marker was useful in screening for other diseases. So then that was in 1979. So this is a more recent study. It's from Metabolites 2022. And they're looking at an association between gut microbiota abundance, so the abundance and the diversity from stool samples, and then measurements of urine metabolites. And it was from almost 1,000 participants. It's the SHIP study. So multiple studies have revealed the association with reduced alpha diversity. So less diversity in the gut is associated with disease phenotypes, with gastrointestinal disorders, obesity, diabetes, so a lot of things. So that's why they looked at that. So when they looked at it, the 4-hydroxyphenylacetic acid was negatively associated with species richness and alpha diversity. So it's somewhat primarily determined maybe by host factors. So that's going to have a significant impact, how much protein and things they're eating. But the higher the 4-hydroxyphenylacetic acid was, the less species richness there was. So there are several important opportunistic pathogens, as I noted, that can produce this compound, such as C. difficile and Pseudomonas. So I'm going to look at it in a test. I'm also probably going to look at Pseudomonas. So the 4-hydroxyphenylacetic acid is considered by some as a metabolic marker of dysbiosis. So I'm going to see what's going on there. So then, not just that. So as I mentioned, there's other markers that can contribute to that. So I need to pull back and look at a full picture. The 4-hydroxyphenylpyruvic acid, which is part of this tyrosine breakdown, generally is always going to be converted to homogenistic. And from homogenistic, it goes to fulvic acid, goes into the Krebs cycle. But if there's a buildup, some of this 4-hydroxyphenylpyruvic acid has been found to have a microbial metabolite. Escherichia converts some of it to 4-hydroxyphenylacetic. So then I'm going to be looking at both of those when I'm looking at total gut bacteria, and do I think there is an overgrowth? I'm also going to be looking at phenylacetic acid. So phenylalanine goes to tyrosine, and that is generally irreversible. Phenylalanine will keep going to tyrosine. If phenylalanine gets very elevated, it can slow down the phenylalanine hydroxylase enzyme. So it's kind of like, that's too much, let's slow it down. So if that gets slowed down, there are some other breakdowns. So normally phenylacetic acid is a minor quantitative marker. It's primarily a metabolite of excess phenylalanine. So that's why it's listed here. So I can see that, is there excess going on? So this is high, this is starting to build up. If phenylacetic acid is around a long time, it can also be of bacterial origin. So it's been positively correlated when it's high with Crohn's disease, and negatively correlated with Faecalibacterium prodznitzi. So I'm looking at all these markers in a group so I can see them all together. This was an interesting study, and they looked at, so when I'm looking at phenylalanine, I'm thinking, well, what about people with PKU that are going to exclude all phenylalanine? Is that necessarily so different? And so when they looked at the specific dietary intake and then the abundance of each of the phyla in different groups. So you can see their protein intakes aren't that different. They're in grams, and actually the PKU people have a little bit more, but they're going to have significantly less phenylalanine, and the control group has normal. But where they made the switches, they have significantly more carbohydrates. And then the fat went way down. So you can see when you look at, they have significantly more Bacteroidetes. So in this type of diet, it's significantly more Bacteroidetes and less Firmicutes. So we have seen something like that before. PKU adults also tended to have higher Akkermansia and Prevotella. And they're also going to be getting not just, more carbohydrates, they're going to be getting more simple sugars, fructose, glucose. So that can also have an impact. Besides phenylalanine and tyrosine, disordered tryptophan can produce indole acetic acid. So the majority of tryptophan from ingested protein is absorbed in the small intestines, and then a certain percentage can still reach the large intestines where it's degraded by a range of commensal microbes. So unabsorbed tryptophan in the colon can be metabolized to indole and indole derivatives. And one of those is indole acetic acid. And so it's a pretty big player. Absorbed tryptophan is going to go to make serotonin and kynurenine. So tryptophan elevation in the gut is due to increased intake or impaired breakdown. So of course I'm going to check diet and I'm going to check digestion. The level of indole acetic acid production is also dependent on the gut bacteria. So once indoles are absorbed from the intestine, they're absorbed into the blood, they get metabolized in the liver, and then they're excreted in the urine. So a change in tryptophan metabolism can result in a negative impact on the diversity of the intestinal microflora, and that can affect the immune system. And that's why there's a lot of research on tryptophan. I think it's also interesting, in 1897, they found that tryptophan was related to indoles, and they attribute it to Bacillus coli, which is now Escherichia coli. That's your history lesson. So this is the three main pathways for tryptophan, for dietary tryptophan to take. If you've seen me talk before or talk to me, you know that I love this pathway. So dietary tryptophan can take two pathways. It can go and it can just be there, and microbiota within the gut can grab it and it can convert it to several different indoles, and one of those being indole acetic acid. You'll see indole acetic acid also written as indole 3 acetic acid or IAA. So it's written a couple of different ways. If the tryptophan gets absorbed by the intestinal cells, it can go to make serotonin with this tryptophan hydroxylase enzyme. And one of the breakdown product of serotonin is 5-hydroxyindoleacetic acid, which can be abbreviated as 5-HIA or AA. Sometimes it has two A's. The other route that it can take is down the kynurenine pathway, and its goal is to make niacin. That's kind of why you have that kynurenine pathway, and that's TDO. But if there's any inflammation, this IDO enzyme, indolamine 2,3-dioxygenase, gets pumped up, and so a lot of that is going to be taken over there. The problem, with that is that it can be related to inflammation. It's generally TNF alpha that's going to increase that IDO activity. And interestingly, the gut microbiota has a critical role in stimulating IDO activity, like who's there can stimulate it. IDO inhibition has been shown to preserve the intestinal barrier, and decreasing IDO can allow for an increased aryl hydrocarbon ligand. So we can get into that too. So this pathway, you can kind of give a better idea of what's going on where. So what's interesting about this pathway is it identifies the microbial pathways. Their pathways are in blue, and the host pathways. So if you think of what's going on in your gut, there's some things you can do that your bacteria can't, and vice versa. So when you look at tryptophan, you can take it to tryptamine in some products, but most of these indole products within the gut are just going to be done by the microbes. so making that indole acid, as opposed to tryptophan going to 5-HTP and to serotonin. Both we can do that and the gut microbes, but only we can take serotonin and turn it into 5-hydroxyindoleacetic acid or melatonin. As far as taking tryptophan to kynurenine and 3-hydroxykynurenine and quinolinic acid. So the microbes can do some of that, but really not much. So they can make kynurenine, and this is kyurenic acid. We make enterocyte acid, quinolinic acid, and picolinic acid. So we can do these things. And the little dots next to them are identifying what they can do with more immune activity. So the aryl hydrocarbon ligands, PXR, and NMDA activity. So we can look at that. When we look at the indoleacetic acid, at least when I look at it, I'm going to look and see, what is indoleacetic acid? It seems to be right there, kind of in the middle. And what are those key bacteria, bifido and lacto, can produce indole intermediates? Akkermansia mucinophilia has some association, but they haven't really parsed all that out. And then I'm going to look at what's going on with tryptophan. Is that tryptophan getting absorbed? Looks like it's getting all pulled. So looking at tryptophan in plasma is going to give me an idea of what's going on over several weeks. So either they're not getting much tryptophan in their diet, or it's getting pulled down to the kynurenine pathway. And they're getting so little tryptophan that they're not able to make much serotonin because there's minimal turnover here. So when I'm looking at this, the aryl hydrocarbon receptor activation is associated with multiple diseases. So the AHR activation, it can increase regulatory mechanisms like IL-10, IL-22, prostaglandin, besides the production of antimicrobial peptides and epithelial repair. So the aryl hydrocarbon receptor is a transcription factor, and there's a lot of research going on with it right now. It was originally thought to function primarily as just a sensor of xenobiotics or toxins, so the P450 activity. The most notable of those toxins was aromatic aryl hydrocarbons, and that's how it got its name, but it's really gone way beyond that. So AHR is activated or deactivated by a number of endogenous and exogenous compounds. So commensal bacteria or their metabolites, like indoleacetic acid, can activate the receptor, contributing to the restoration of gut normobiosis and homeostasis. Lactobacillus reuteri, Lactobacillus bulgarius, indoleacetic acid, and kynurenine can all regulate that inflammation with AHR activation. There's also AHR activation binding with natural, like polyphenols and flavonoids. So it's got a lot going on that tryptophan is a big player in. So this is a nice diagram. I left the mice in here so you can see it was a mouse study. They had pregnant mice and mice with metabolic syndrome. And they also had some fecal matter from people with metabolic syndrome and pregnant women. So mucosal inflammation, the activation of this IDO activity, so it's the same IDO that I had mentioned, can hijack tryptophan. Like we saw that tryptophan was really low and the kynurenine was high. So when it comes into the gut lining and this IDO just grabs it and pulls it down this kynurenine pathway. So it leaves less for these indoles to be made. So there's, eventually some altered gut microbiota. There's a decrease in this aryl hydrocarbon agonist production. And then there could be an increase in IL-22 and some low-grade inflammation. There's also not going to be as much to go over to the serotonin pathway. So mice who were bred to miss this IDO activity didn't have any of this disruption. So looking at this pathway, it was an article about a website. And it's a website all about tryptophan, everything you ever wanted to know about tryptophan and its metabolites called the tricats. And they looked at different things, but the estimated odds ratio for the prevalence genes in production to bioactive indoles. So I have this indoleacetic acid, but this was a big table that looked at all the different indoles and who could be the predictors of those. So desulfovibrio, while it doesn't produce it, was more predictive of a higher indoleacetic acid. So it may be that type of gut that's going to support that's going to support a more indoleacetic acid, while enterococcus was generally opposite. So this panel is looking at like a healthy adult and their tryptophan breakdown, and sadly, an unhealthy adult. So in, panel A, everything's going right. There's good gut microbiota. There's not an increase of the kynurenine pathway. And so things look okay. But in this other pathway, there's some dysbiosis in the gut. There is some inflammation. There's a decrease in these AHR ligands. And so there's an increase in kynurenine in that IDO activity. So in this kind of situation, I'm looking at the tryptophan, the 5-hydroxy, which those don't look as bad here as the other one, but it looks like there's some inflammation and that kynurenine's getting pulled, tryptophan's getting pulled down to kynurenine. So eventually it could lead to lowering of that tryptophan level. This person also has, but they don't have inflammation. They do have some increased gut permeability. And that indoleacetic acid is a little bit on the lower end. So indole is a signaling molecule for bacteria, for influencing motility, biofilm formation, antibiotic resistance, and virulence. And it's been shown to inhibit colonization of pathogens like salmonella. But there is indole overproduction, and that can increase its export to the liver, where it gets sulfated, and it can be a uremic toxins for the kidneys. Plus its effect as a signaling molecule, both for bacteria and the intestinal cells, is dependent on its size. So if the concentration gets too high of this indoleacetic acid, it can kind of decrease its activity. So the other thing that I'm looking at when I'm looking at this whole tryptophan and all aspects of it from the gut and everywhere else, I'm looking specifically at that KT ratio because it's known to identify IDO activity, can be a marker of inflammation. It's also been related to severity of IBS and is related to IBD. In some cells, in another study, it was correlated negatively with glutathione use. So if I see somebody with a really high KT ratio, I'm generally going to look and see, what's the glutathione status look like? Plasma cystine has been considered rate limiting for glutathione production. Alpha hydroxybutyric acid can identify glutathione, how much is being produced. And pyroglutamic acid is identifying that you're trying to make it, but you don't have everything. So most clinicians will add glycine or NAC if that's elevated. Additionally, I'm going to look at the 5-hydroxyindoleacetic acid. This is, from Stat Pearls 2022, and it has a list of all the things that urinary 5-hydroxyindoleacetic acid can be related to. So it can be related to endocrine tumors, which we don't see too often, malabsorption, IBS, especially IBSD compared to controls, and in celiac disease. So this last pathway is looking at serotonin in IBS. So this is, the influence of gut dysbiosis on serotonergic signaling. So this is looking in this pathway. You have a diet, you get tryptophan. If your tryptophan goes into a gut and you have a high level of microbial species that upregulate serotonin production, essentially the enzyme that takes tryptophan to serotonin, you're going to have more of serotonin within the gut, and that's associated more with IBS-D, low-grade inflammation. If you eat and your tryptophan goes into a gut that has higher levels of gut bacteria that downregulate 5-HTP, you're going to have less serotonin, and that's generally associated with IBS-C or constipation. And some of these bacteria that up or downregulate are known. So here's another one. So I'm going to move from tryptophan to citrulline. So low plasma citrulline may identify impaired amino acid absorption. In a review of patients with diagnosed bowel disorders compared to control, decreased blood citrulline was associated with impaired enterocyte function and small bowel absorptive capacity. And that was a nice big study with like 500 people. So plasma citrulline was also correlated with some transporters, and I'm not going to go into that detail. But citrulline is mainly synthesized by glutamine as a precursor substance in the intestinal mucosa. When the epithelial cells in the intestinal mucosa are damaged, the synthesis of citrulline is significantly reduced. So this study found that citrulline is an amino acid involved in intermediate metabolism. It's not incorporated into proteins. So it's mainly produced by enterocytes. So if there is in the small bowel, so if there is any issue with that, then you're going to see lower levels. So it's considered a reliable surrogate marker for small bowel absorptive capacity. And then another one I look at, and if you've talked to me, you know I look at this for some other stuff too, is glutamine to glutamic acid ratio. And that's been proposed as a biomarker for obstructive bowel disease, distinguishing it from obstructive bowel disorder to short bowel syndrome. So this glutamine to glutamate ratio, higher is always better. The Framingham study and the PRIMED study have also found higher levels to be related to lower risk of metabolic and cardiovascular disease over decades. So it's not just for this. But I usually, if I'm looking at this along with a gut picture and these might be concerned, I'm going to look at D-lactic acid because it can be elevated if somebody truly has short bowel syndrome. And remember, glutamine is metabolized to citrulline. So one of the things I'm looking at is D-lactic acid can be a complication of short bowel syndrome. So that's something to look for, and the specific bacteria. Additionally, but usually that's low. But another way you can get more D-lactic acid is the methylglyoxal pathway, or just methylglyoxal. So methylglyoxal comes from glycation, from advanced glycation end products, or AGE, that are associated with metabolic impairments and oxidative stress. So D-lactic acid levels serve as a reflection of cumulative MGO exposure. So when you have MGO, it goes through this glyoxylase defense system, which uses glutathione to make D-lactic acid. So that's why it's representative of it. You can also just ingest D-lactic acids in some forms. Another marker that I like to look at is plasma proline and changes in gut bacteria and intestinal permeability. This is another nice recent study, and they looked at that in relationship to depression. So when they're doing this multi-omics approach, they look at the microbiota gut-brain axis in depression, and that demonstrated that the microbiome-dependent elevations in plasma proline were significantly associated with depression severity in humans. And then when they looked to mouse studies, they found that if you supplemented proline to depressed mice, it exacerbated their depression. So the authors in the study proposed that diets with reduced proline content may have an impact on ameliorating depressive symptomology. And they have a nice diagram here. So here's this diet and the proline content. It goes into the gut, and the impact of it is going to be dependent on what's in your gut. So a gut bacteria that has more of these, Bifido, Collinsellia, Roseburia, is going to do better. It's going to lessen depression severity. But a gut bacteria that has more of these, the Enterobacter prevotella, may increase depression or is associated with increased depression. So when I'm looking at these as a whole, I'm going to look at plasma proline and see, like, this would worry me if this were somebody who was depressed. So plasma proline, it has been positively associated with species from the Bacteroids like Bacteroides phylum and Prevotella genera, but it's negatively associated with Bifido and other short chain fatty acid producing bacteria like Roseburia. So I'm going to want to look at this, like look at that low Roseburia and that super high proline. That would be of concern to me. They also found that there was serum zonulin levels correlated positively with dietary proline consumption. So if you have more proline, you're more likely to have a higher serum zonulin level. So also hyperprolinemia has been associated or linked to epilepsy, schizophrenia, seizures, and impaired cognitive functions in humans. And there are studies of patients who, despite consuming a high proline levels, had low circulating proline, they have a high proline intake, but they had low circulating proline levels. They had a different microbial composition that was consistent more with the people who had lower depression scores. So a high proline diet may not be for everybody, and you may have to look at it based on an individual. So when I'm looking at, this protein as a whole, I think this is like the back of a report, and this person had, all their amino acids except these few that were elevated. So that would be a concern for me. So what I'm looking at is these things to kind of help these out. I want to reduce protein delivery to the colon. That should be something you might think of for everybody. Is there a change in GI physiology? Don't stay on antacid therapy. It can make the stomach more alkaline, obviously, but also can aging. So you're going to see that more in older adults. An increase in intestinal transit time. So somebody that has diarrhea may have a problem. So they may also have more mucosal disease. For a lot of people, just decreasing total protein intake can decrease the percentage that goes to the distal colon. Increasing digestibility. So that can be protein source. So some are better digested than others, as well as cooking method. So prolonged heating may reduce that. And then I would also look at, digestive enzymes and how that's going. And then adjust other food factors. So there's, other things that can, inhibitors. Reduce specific pathogenic catabolites. So maybe don't focus as much on the sulfides or the phenolic compounds or the indoles, especially if you've tested and you've seen those to be elevated. Maybe pull back on those contributing proteins until you can work on the gut. Encourage protein synthesis and fermentation. So promote carbohydrate fermentation throughout the bowel. So protein, you're going to have less protein showing up to be available in the colon if there's also a lot of carbohydrates, specifically undigestible carbohydrates like fiber. So, they have this, combined slowly and rapidly fermented fiber. That's one of their suggestions, wheat bran plus resistant starch. And then modify gut microbiota. So aim for less fermentive potential and more reduced specific catabolites. So I think these are all the things, obviously I don't think many people are doing antibiotics, but probiotics, prebiotics. There's also products now that you can get. They're like phytobiome. They're phytochemicals that are specifically focused for the gut microbiome. So I would think of those things. The other thing is, like, I'm going to check digestion. It's plasma amino acids are better markers of just overall protein intake and the whole digestion absorption process. Urine is going to sometimes correlate, as it does here, but it can be pretty impacted by 24 to 48 hour intake. But if it's extremely elevated, it can tell me that there could be an impairment somewhere. It can also tell me if a lot's being spilled in the urine. But getting both of them can certainly give a lot more information. And then I'm going to look at just digestion. Is there fecal fat? Because that's going to give me some information. How's the elastase? This looks fine. So then beyond all those is looking at pancreatic elastase. The study was pretty interesting. It was 2021. And they looked at the effect of exocrine pancreatic function on intestinal microbiota composition and how it alters that whole microbial derived metabolites. And they looked at if there was low fecal elastase or low elastase, that you're going to have, it was associated with increased arginine, threonine, serotonin, which I'm looking at with 5-HIA, and decreased acylcarnitine species from medium to long chain. So there could be some impact on energy generation through impairments in beta oxidation. So I'm going to look at those. Another aspects, so this is going beyond proteins, so we're moving on from proteins, and we're looking at polyphenols. So phenolates are anions, salts, and esters of phenols, sometimes called phenolics. So phenolic compounds are classified as simple phenols or polyphenols. And so it's based on the number of phenol units. Phenols are synthesized in industry or made by plants or microorganisms. So polyphenols are going to be abundant in plants and structure, and they're going to get to the gut. So a large portion of unabsorbed polyphenols make it to the large intestines, where they interact with colonic gut bacteria and that are capable of breaking it down and degrading those compounds. So here are a few that they're big in the research, and I'm looking at those. So 3,4-dihydroxyphenylpropionic acid. It's primarily it's formed from microbial transformation of dietary polyphenols. It can also come from metabolites like dopamine, phenylalanine, tyrosine, and tryptophan. So those are phenolic compounds, so they can get kind of caught up in this. But just like the breakdown products that we looked at for phenylalanine tyrosine breakdown can be impacted by polyphenols, this can also be impacted by these compounds. But primarily 3,4-dihydroxyphenylpropionic acid is going to be a breakdown from polyphenols. So the big one, the microbiota that they've found are Clostridium, Escherichia, Bifido, Lactobacillus, Eubacterium, and those are the ones that they've identified. And the fermentation substrates or phenolics, and this tends to focus more on flavonoids. This compound does correlate with homovanillic acid pretty strongly, so I generally look at that. I'm not going to just make a judgment on catecholamine turnover without also looking at some of these compounds. So another compound I'm looking at is 3,5-hydroxybenzoic acid. So that really looks more at total gut microbes, so that's why I'm looking at the phyla. It is primarily from a compound in whole grain, in whole wheat, rye, barley kind of sources. And I generally don't even try to pronounce this compound, but gut bacteria will act on it and make this 3,5-dihydroxybenzoic acid. So in population studies, it's been used as a biomarker of whole grain intake. In some cell studies, they've looked that it does have antimicrobial activity against E. coli, Pseudomonas aeruginosa, Staphylococcus aureus, albicans, so kind of looking at those. And then the last one I'm looking at is 4-hydroxybenzoic acid. So its main, bacteria are Clostridium, Eubacterium, and also just total. They're phenolates. They come predominantly from anthocyanins, so those purple foods. That's why my berries. But they also can be impacted by endogenous metabolites like the dopamine, phenylalanine. So it's generally found in certain purple foods. But 4-hydroxybenzoic acid has been shown is one of 3 urinary metabolites, along with histamine and azelaic acid, that was able to discriminate between a low FODMAP and a high FODMAP diet. So it was found to be, it increased when when people were on a low FODMAP diet. And it was found to be positively associated with Firmicutes and Akkermansia muciniphila, and negatively with bifidobacteria. So another compound I look at is benzoate. So this is the benefits of benzoate in animal models. I mean, our digestion is pretty similar to pigs, but I think it's important to know that excess benzoic acid is known to have serious implications, because benzoic acid should get bound with glycine and get excreted. So move over to glycine, to hippurate. So hippurate's really kind of representing how much benzoic acid. So this same SHIP study also looked at benzoate and hippurate, and that they found certain gut bacteria associated with higher levels of benzoate. So the big category here, this proteobacteria, which includes a wide variety, but Escherichia, Salmonella, Vibrio, Yersinia was associated with higher levels of benzoic acid. But when you look at the hippurate, you see that hippurate is positively associated, unlike the 4-hydroxy marker we'd seen from tyrosine. So as hippurate goes up, Shannon diversity goes up. As hippurate goes up, species richness goes up. There's also studies looking at increased hippurate being associated with a Mediterranean diet. And then this study was pretty interesting. They tested the hypothesis that just having a polyphenol-rich diet can decrease serum zonulin levels and beneficially alter intestinal microbiota, which they did. They found that to be true, that a polyphenol-rich diet increased fiber fermenting bacteria and butyrate producing bacteria such as Faecalibacterium, and reduced zonulin levels. So when I'm looking at these, I'm looking at, are these polyphenol markers all really low? Like, that's a little concern. I might look into that. Or are they really high? If they're really high, I'm going to look at what kind of diet do they have and what's their gut bacteria look like. If they're all really low, I'm going to look at the same thing and just kind of see that balance. So another thing to look at is, we look at kidney function, urea cycle. There's a lot of research looking at the microbiome and metabolomics with kidney health. And those are things that you can look at that can have a significant impact. So slow intestinal transit, impaired protein assimilation, which makes sense, decreased fiber. So it's good to look at all these, in relationship to kidney health. Now, this is a last one, so I'm speeding up here, but this is a great article, and it looks at microbes involved in vitamin synthesis. So these are the vitamins and these are the microbes. And then if they're deficient, what effects they have on the intestines. So when I'm looking at vitamin need, so I look at this B complex, since it's difficult with metabolomics for most of these B complex vitamins to look exactly. There isn't a known pattern for these, but there is some research looking at these dehydrogenase enzymes. If they're lacking the B vitamins, these markers become elevated. Here we just see pyruvic acid is elevated, so it's not across the board. But I'm also going to start looking at, are these B complex available, and what bacteria should I look at? So Bacteroides, Prevotella, and desulfovibrio can produce thiamine, B1. So I'm going to look and see, are those a lot there? I'm not saying I want to increase or decrease these, but what impact they can make. Fusobacterium, Proteobacteria, Prevotella are going to do B2. So I'm looking at each of these bacteria and the total. And so this is one that I've seen. When you look at these, you'll see that this particular pattern looks pretty significant. So somebody who they're missing one or more of these B vitamins, the LA is lipoic acid. And so none of these are really working well. So they're missing one or more of these B vitamins, probably the whole, as a complex. And so this dehydrogenase enzyme, which is known to be impacted, isn't going through. So you can have decreased abundance of Peyer's patches and B cells, which can be insignificant. You could have impaired intestinal cell growth. It's associated with relapsing IBD. And then folate also is going to be impacted. So it's got specific bacteria that produce it, and it's got a pretty long list of impairments when it's inadequate. There's also there's research looking at fructose and the pattern, that it can play in negatively influencing the heart-gut axis. So generally, just getting fructose is not going to have an impact. But if you get concentrated sources like a soda, specifically if you're not eating stuff, that can have an impact. So increased fructose intake has been known to increase gut barrier permeability, inflammation, which makes sense. People getting a lot of fructose probably aren't eating the best diet, but that has been seen in research. There's also markers associated with IBD. And different types of IBD. So Crohn's disease compared to ulcerative colitis. Citric acid tends to be low in both of those. Alanine usually is a little more elevated. It's also elevated in metabolic disease. Hippuric acid. Hippuric acid is, and just like we went through with benzoate, generally lower. So you might not be converting that benzoate over, and you might have higher benzoic acid in the intestines. Cysteine was found to be lower in Crohn's disease. And then pyroglutamic acid was found to be higher in both. So there could be an impairment in glutathione production. And then research has also found low plasma histidine associated with both Crohn's disease and ulcerative colitis. So this is my last slide, that there was a significant positive correlation found between styrene levels and disease activity in ulcerative colitis. And the point really is there's not much that you're being exposed to that isn't also impacting the activity of the gut microbiome in some way. So that is the end of my presentation. And so I think I'd be ready for any questions if there are any.
Our first question, what natural substances can increase AHR?
It's not really increasing alone. It's really balancing. So balancing the gut microbiota, like knowing what the gut microbiota is and trying to get more of those good bacteria and decreasing those. Balancing digestion. If somebody has increase of urinic acid or a high KT ratio, I'm going to look and see where's that inflammation. Do they have any impairments in digestion? Are more of those compounds reaching the gut? So it's not just stimulating because it has some good qualities and some, bad, not necessarily bad qualities, but, you want to, so balancing the microbiome and digestion and getting a good diet, kind of those same things you might always do for good health.
Thank you. Our next question, what are the amino acids related to GI motility?
I don't know if there's any directly related. There's a lot of factors that come into play with GI motility. I think if GI motility is impaired, you can have more of those amino acid substrates. I mean, motility at all, like, if somebody gets more fiber in their diet, certainly they're going to have better motility. So just making sure their gut bacteria is ready for increase in fiber and then just slowly increase those. That's going to help with gut motility. The same things you would always think of if somebody was constipated: more fiber, maybe supplemental fiber, kind of be careful how you increase that depending on their gut microbiome makeup, more fluids, all those kind of things. So I wouldn't consider like just a specific protein as having that impact. The protein is impacted, makes an impact when the motility is increased or really decreased.
Thank you. We've had a few people ask this question. This will be our final question. But basically, is it better to do plasma or urine amino acids?
Plasma is really what's related to dietary status, like your function. If you were going to do, a research study on how, are people eating and digesting protein, you would likely do plasma. It's going to give you more of a long-term intake. Urine is more impacted by the day-to-day variation. It can, be reflective of 24 or 48-hour intakes. So it can be similar if your diet's really the same. You wouldn't want to, suddenly have a steak the night before and you never do that. That's going to make an impact. But so I generally prefer plasma, but there's a lot more research coming out now on urine. So they both give you a picture of what's going on. It's just if you do plasma, look toward the research that assesses plasma. And if you do urine, look at the research that has assessed urine markers.
Thank you very much. That will be our final question. Thank you all and have a great rest of your day.